So, hello class, very good evening. So, today is the day. Sorry, just let me check what is the notifications here. Today is the day we will deal with the two topics. That is, I check it later. That is, mm, ultrastructure of cell and chromosomes. So these were two classes, but tomorrow I will be off. I have to go out of station, so I cannot take tomorrow your class. So today I will be taking your class for tomorrow also, so that we can compensate with that. So on that note, it's already 8. We can start with the ultrastructure of cell and organelles and their functions. So let's just wait for one more minute, then we start. Student, I just got a call. Just wait for a second. Uh, it's an important call I have to take. I hope you're still with me, everyone. Just wait for a second. Hello. Sir, call आई थी सर आपकी. सर सिमरन. हाँ जी, हाँ जी. हाँ जी. नहीं नहीं सर. हाँ जी, हाँ जी. सर मैं आपको सर एक घंटे में कॉल करूँ. एक घंटे में कॉल करूँ सर. आपको मेरी क्लास करनी है ऑनलाइन. मैं आपको कॉल करता हूँ. हाँ जी सर. हाँ जी, हाँ जी सर. मैं एक घंटे में आपको कॉल करता हूँ सर. ओके सर. मैं करता हूँ सर. ठीक है सर, ठीक है. ओके. So class, let's start. Ultrastructure of cell and cell organelles and their functions. So I will be dealing with the unit 1, that is ultrastructure of cells and their cell organelles and their functions. So we will be dealing with the all parts of from the cell wall to protoplasm, cytoplasm, endoplasmic reticulum. So all the structures we will be dealing in this case of your today's lecture. So this is a plant cell. You can see that vacuole. So here we have let's start from the chloroplast. We have a mitochondria, we have cytoplasm, rough endoplasmic reticulum, nucleus, nucleolus, nuclear envelope, cell wall, smooth endoplasmic reticulum, ribosomes, plasmodesmata, Golgi apparatus, peroxisomes, plasma membranes. So all these parts belongs to your ultrastructure of your cell. So cell organella is a specialized entity which is present inside a particular type of cell that performs a special functions. So there are various cell organelles, out of which some are common in most type of cells like cell membranes, nucleus and cytoplasm. However, some organelles are specific to one particular type of cells like plastids, cell walls and plant cells. So as per the requirement, each cell has their own cell organelles. In some they are included, in some they are not. For example, in plant cells, we have plastids and cell walls in the case of plant cells. So this is the cell organelles of difference between your plant structure and animal structure. So here we can see very nice difference that it has in the plant structures. It has mitochondria, lysosomes, Golgi vesicles, Golgi apparatus, ribosomes, ribendoplasmic reticulum, nucleolus, nucleus, cytoplasm, intermediate filaments, vacuole, vacuole membrane, chloroplast, smooth endoplasmic reticulum, peroxisome, amyoplast, cell membrane, cell wall, granules, microtubules. So these all are included in the plant cell structure. Whereas in the animal cell structure, you have a cell membrane, you have a chromatin, you have a nucleolus, then there's a nucleus, then there's a smooth endoplasmic reticulum, then there's a Golgi apparatus, then there's Golgi vesicles, cytoplasm, lysosomes, microtubules, centrosomes, vacuole, ribosomes, rough endoplasmic reticulum, peroxisomes, mitochondria. So we will discuss mainly all the basic parts today of your animal structure to the and plant cell structures also. So all parts will be taken care for today. So let's start with the cell membrane, that is plasma membrane and plasma lemma. So plasma membrane, which is present at the cell surface of the animal cell structures mostly. So this is how it looks like a hydrophilic case. So plasma membrane is composed of lipids and proteins where the composition might fluctuate based on the fluidity, external environment, and the different stages of development of the cell. So structures are, it consists of phospholipid bilayer along with two types of proteins with embedded proteins and peripheral proteins that functions in providing shape, allowing the movement of the particles in and out of the cell. The most abundant lipid which is present in the cell membrane is a phospholipid, which contains a polar head group attached to the two hydrophobic fatty acid tails. The embedded proteins act as channels for the transfer of particles across the cell, with some proteins acting as a receptor for the binding of various components. The peripheral protein function as to provide fluidity as well as the mechanical support to the structure. So what are the functions of plasma membrane? It provides mechanical support that facilitates the shape in the cell while enclosing the cell, and it's a component from the external environment. It regulates what can be allowed to enter the and exit the cell through the channels, acting as a semi-permeable membrane which facilitates the exchange and essential compounds are required for the survival of the cell. It allows the interaction between cells required during the tissue formation and cell fusion. So that was something about cell membrane. Just a second. So now let's come the next part, that is cell wall, which is present after the cell membrane, which is present in both cases of your cell, plant cell and animal cells, both cells it is present. So students, this is very basics that what we are doing today. I hope you have done this in your plus 2 class, but still it's a part of our lectures. So I felt like to teach you each part in details today, so that we are on the good kind of good platform. We are all on the right platform then. So here the plant structure wall, it has pectin, cross-linking glycans are there, cellulose microfibrils are present. Then there is a middle lamella, primary cell wall is there, then a plasma membrane. So this is how your looks like. And there's a two type of cell walls, gram positive and gram negative. So gram positive has peptidoglycan and gram negative bacteria has outer membrane as this one. It has cell wall there. So gram positive bacteria and gram negative bacteria. So it has small peptidoglycan, but it has on two sides the plasma membranes are present. So it has also have outer membrane in the gram negative. So cell wall, it's an additional non-living layer present outside the cell membrane in some cells that provide structure, protection, filtering mechanism to the cell wall. Uh, In a plant cell, the cell wall is made up of cellulose, semicellulose, and proteins, while in fungal, it is composed of chitin. A cell wall is a multi-layered with a middle lamina, a primary cell wall, and structural secondary cell wall. So, middle lamina contains polysaccharides that provide adhesion and allows binding of the cells to one another. And after middle lamina is the primary cell wall, which is composed of cellulose. The last layer, which is not always present, is the secondary cell wall made of cellulose and hemicellulose. Its function is to protect and maintain the shape of the cell. It also helps the cell withstand the turgor pressure of the cell. It initiates cell division by providing signals to the cell and allows the passage of some molecules into cell while blocking others. So there is a centrioles. So here you can see that they are present during your mitosis and meiosis at the edge, which helps in the separation of your chromosomes during mitosis and meiosis. So it has distal end and there's a proximal end, and these are your microtubule triplets. So these are microtubule triplets, and they are arranged in like this pattern, the centrioles. So they are the tubular structures, most fondly in the eukaryotic cells, which are composed mainly of the protein tubulin. And centriole consists of cylindrical structure made with the 9 triplet microtubules that surround the periphery of the centriole, which has center and Y-shaped linker. Further, the functions it's include are including the cell division. Centrioles have crucial role in forming spindles fibers, which assist the movement of chromatids towards their respective sites. They are involved in the formation of cilia and flagella. So cilia and flagella, so they are two type, gram negative flagellum, gram positive flagellum. So here you can see in the gram negative there are 4 rings, in the gram positive there are only 2 rings present. And these are various flagella is there, and these are cilia present in the bacteria. So cilia and flagella are tiny hair-like structure projecting from the cell made of microtubules covered by the plasma membrane. Cilia are like hair-like projections that have 9 plus 2 arrangement of microtubules with a radial pattern of 9 outer microtubule doublets that surrounds 2 singlet microtubules. This arrangement is attached to the bottom with a basal body. Flagella is a filamentous organelle, the structure of which is different in prokaryotes and eukaryotes. In prokaryotes, it is made up of the protein called flagellin, wrapped around in helical manner, creating a hollow structure at the center throughout the length. In eukaryotes, however, the protein is absent and structure is replaced with the microtubules. The most critical role of cilia and flagella is movement. So these are responsible for the movement of the organisms as well as for the movement of various particles present around the organisms. Some cilia present in some particular organs may have the function of sense. The cilium in the blood vessels, which helps in controlling the flow of blood, is an example of that. Then comes our next structure, that is chloroplast. So here in the chloroplast you can see there is an outer membrane, intermembrane space, lipid droplets, chloroplast DNA. We have a lamella, then there is a granum present, and starch granules are there, ribosomes, lumen, thylakoids, stroma, intermembrane. So this is all your chloroplast structure is made up of. So let's see. So chloroplast is a plastic that is involved in the photosynthesis in plants and algae. It contains essential pigments called chlorophyll necessary to trap sunlight for the production of glucose. Its structures looks like a double membrane structure with own DNA which is inherited from the previous chloroplast. These are usually length shaped with the shape number varying according to cells, and they have outer membrane and inner membrane and a thylakoid membrane that enclose the gel-like matrix called stroma. The outer and inner membrane is porous, allow the transport of the materials, while the stroma contains chloroplast ribosomes, proteins, and starch granules. So functions look like it's a primary center for light dependent and light independent reactions during photosynthesis, and different proteins present in chlorophylls are involved in the regulation of a photorespiration. So in photorespiration and in photosynthesis, both in cases your chloroplasts play a major role. So this is students, you're freshen up with your all structural parts, how these structures works, how things are working out. So I try to make it as simple as possible for you, and with the figures, with the structure mainly telling about each structure, what are they and their functions. Just keep them as simple as possible, so that whenever you have these things, so you can just go through this slides and you can know that what is what. And the reason that I have told you, some students are still sending me the assignments, some are not. So the so you can also send me at the end of your thing. So whatever we are learning, so at least you remember them for the later part at the end of your workshop is over, because workshop will over, you will get your certificate, but you will don't remember these things. So that's why I always convince you all and emphasize on the assignment part. So keep that thing in your heart then. So then comes the cytoplasm. It refers to the everything that is present inside the cell except the nucleus. This structure consists of cytosol, a gel-like substance that contain other matter, cell organelles, some smaller cells like bodies bound by separate membranes, and cytoplasmic inclusions. Insoluble molecules that store energy and are not surrounded by any layer. So cytoplasm is a colorless and has about 80% water along with various nutrients required for the cell. It is known to have properties of both viscous matters as well as elastic matter. Under its elasticity, cytoplasm helps in the movement of material inside the cells by a process termed cytoplasmic streaming. So most of the vital cellular enzymatic reactions like cellular respiration, translation of mRNA into proteins occur in the cytoplasm. It acts as a buffer and protects genetic material as well as other organelles from the damage due to collision or change in the pH of the cytosol. The process called cytoplasmic streaming helps in the distribution of various nutrients and facilitates the movement of cell organelles within the cell. Then comes your cytoskeleton, which helps in the movement of a cell from one place to another. So this is how the cytoskeleton looks like. So it's a number of fibrous structures are present in cytosol that helps give shape to the cell while supporting cellular transport. Structure looks like there are three different classes of fibers that is microtubules, microfilaments, and intermediate filaments. So these are separated based on the protein present in them. The critical function of these cytoskeleton is to provide shape, mechanical support to the cells against deformation. It allows the expansion and contraction of the cell, which assist in the movement of the cell. It is also involved in the intracellular and extracellular transport of the materials. Then comes your endoplasmic reticulum, that is ER, which is present outside your nucleus. That is, it has a cisternae. These are your cisternae, and outside the small dots are present are called ribosomes. And then there's cisternae space. In between the cisternae space is called. Then outside one is the rough endoplasmic reticulum, and there is outside of your rough endoplasmic reticulum without ribosomes. They are called as smooth endoplasmic reticulum. So endoplasmic reticulum is present as an interconnection of tubules that are interconnected to the nuclear membrane in eukaryotic cells. So there are two types of ER based on presence or absence of ribosomes on them. That is a rough ER and a smooth ER. The rough ER has ribosomes attached to them and is involved in the protein synthesis. A smooth ER which lacks ribosome and has functions during lipid synthesis. So structure looks like endoplasmic reticulum exists in three forms with cisternae, vesicles, and tubules. Cisternae are sac-like flattened unbranched structures that remain stacked one on top of another. Vesicles are spherical structures that carry proteins throughout the cell, and tubules are tubulous branch structures forming a connection between cisternae and vesicles. ER contains many of the enzymes required for several metabolic processes, and surface of the ER is essential for the operations like diffusion, osmosis, and active transport. One of the crucial functions of ER is the synthesis of lipids like cholesterol and steroids. Rough ER allows for the modification of polypeptides emerging out of the ribosomes to prepare secondary and tertiary structure of the proteins. ER also synthesizes various membrane proteins and has a crucial role in preparing the nuclear envelope after the cell division. So these are your endosomes stages. Early it looks like one, then early stage 2 looks like this, then it's like going out. So also in this case late 1, late 2, and then 1 is fused out. So these are how endosomes works like. So they are the membrane bound compartment within the cell originating from the Golgi networks. There are different types of endosomes based on morphology and time they take for endocytoid materials to reach them. The early endosomes are made with a tubular vesicular network, while the late endosomes lack tubules but contain many closed packed intraluminal vesicles. The recycling endosomes are found with microtubules and are mainly composed of tubular structures. Its main function is endosomes allow the sorting and delivery of internalized materials from the cell surface and transport of material to the Golgi or the lysosomes. So Golgi apparatus, Golgi bodies now comes. So it has in these are incoming transport vesicles are coming up here. So there is a cis phase and this is a trans phase. One with the bulging side is the cis phase, or the one with the non-bulging side is the trans phase. And these are secretory vesicles, lumen, cisternae. Here also cisternae, lumen. This is cisternae space in between is present. So the Golgi apparatus is the cell organelle mostly present in the eukaryotic cells, which is responsible for packaging of macromolecules into the vesicles so that they can be sent out to their site of action. So wherever their work is, they could go there. So structures looks like it's a pleomorphic. However, it is typically exists in three forms: cisternae, vesicles, and tubules. Cisternae is the smallest unit of Golgi complex and has a flattened like structure which is arranged in bundles of parallel fashion. Tubules are present as a tubular and branch structures that radiate from the cisternae and the fenestrate at the periphery. The functions are Golgi has an essential part in the directing proteins and lipids to the destination and act as a traffic police. So they are involved in the exocytosis of various products, proteins like zymogen, mucus, lactoprotein, and parts of thyroid hormone. Golgi complex is involved in the synthesis of other cell organelles like cell membrane, lysosomes, among others. And they are also involved in the sulfation of various molecules. Then comes your lysosomes. So there are enzymes present and proteins are present, and outside is the membrane of your lysosome. So lysosomes are membrane bound organelles that occur in the cytoplasm of animal cells mainly, and they organelles contains an array of hydrolytic enzymes required for the degradation of various macromolecules. So there are two types of lysosomes: primary lysosomes containing hydrolytic enzymes like lipases, amylases, proteases, nucleases. Secondary lysosomes are formed by the fusion of primary lysosomes containing engulfed molecules of organelles. So structure looks like the shape of lysosomes is regular or pleomorphic. However, mostly they are found in spherical or granular structure. Lysosomes are surrounded by a lysosomal membrane that contains enzymes within the lysosome and protects the cytosol with the rest of the cell from the harmful action of the enzyme. So functions are these organelles are responsible for intracellular digestion, where the large macromolecules are degraded into smaller molecules with the help of enzymes present in them. Lysosomes also perform the critical function of the autolysis of the unwanted organelles within cytoplasm. Besides these, lysosome is involved in various cellular processes including secretion, plasma membrane and cell signaling and energy metabolism. Then comes your mitochondria. So here the mitochondria looks like it has cristae, outer membrane, inner membrane, matrix, inner membrane again. The DNA is there present and the ribosomes. That's the basic structure of a mitochondria. So mitochondria are double membrane bound cell organelles responsible for the supply and storage of energy of the cell. That is, they are the ATP of the house. Primary purpose is to release ATP, adenosine triphosphate. Structure contains two membrane with outer layer being smooth while inner is marked as a with foldings finger-like structures called cristae. And inner mitochondrial membrane contains various enzymes, coenzymes, and components of multiple cycles along with the pores for the transport of substrates, ATP, and phosphate molecules. Within the membrane is a matrix that contains various enzymes of metabolic process like Krebs cycle. Uh, Mainly function, as we have discussed, they are the synthesis of energy, functioning for all the cell organelles, helps the balance of calcium ions. It's in the liver have the ability to detoxify ammonia. So these are the various functions of your mitochondria. Then comes the nucleus, which is a double membrane bound structure responsible for controlling all cellular activities. One of the largest organelles with 10% of total space in it. It is often termed the brain of the cell, or as it provides commands for the proper functioning of the cell organelles. The nucleus is clearly defined in the case of eukaryotic cells. However, it is absent in prokaryotic organisms with the genetic material distributed in the cytoplasm. So this is your nucleus, nuclear pore, chromatin, nucleolus, nucleoplasm, nuclear envelope. These are the structures all looks like. So in the structure part, the nucleus consists of nuclear envelope, chromatin, and nucleolus. It is similar to the cell membrane that is found in structures and composition, and it is movement of proteins and RNA in and outside of the nucleus is happening here. Chromatin in the nucleus contains RNA or DNA, which are your nuclear proteins as a genetic material responsible for carrying one genetic information from one generation to another. And nucleolus is a nucleus within the nucleus. It is a membraneless organelle that is responsible for synthesis of ribosomal RNA and assembly of ribosomes required for the protein synthesis. So it's act as a genetic information, transfer of genetic information is done, transcription is done from mRNA molecules, and nucleus controls the activity of all other organelles while facilitating cell growth, cell division, and synthesis of proteins. Then comes your ribosomes. It has amino acids. These are your ribosome large subunit. There is a transfer RNA, there is mRNA, and a ribosome small subunits. So ribosomes are the ribonucleoproteins containing equal parts of RNA and proteins along with an array of other essential required for protein synthesis. So in prokaryotes, they exist freely, while in eukaryotes they are found in either free attached or endoplasmic reticulum. The structure looks like it has two subunits. In the case of prokaryotics, they have 70S with large subunit and 50S and other small 30S subunits. In eukaryotics, 80S ribosomes with 60S large subunit and 40S smaller subunit. Ribosomes are short-lived, as after the protein synthesis, the subunits split up and can be either reused or remain broken up. Ribosomes are the site of biological protein synthesis in non-living organisms. They arrange the amino acids in order indicated by transfer RNA and assist in protein synthesis. Now comes the vesicles. So they are on the left side is a liposome, on the right side is dendrimersome. The blue part of the molecules are hydrophilic and the green parts are the hydrophobic. So vesicles are the structure present inside the cell, which are either formed naturally during the processes like exocytosis, endocytosis, or transport of material throughout the cell, or they might be artificial, which are called liposomes. So structure containing looks like it has a liquid cytosol, which is enclosed by the lipid bilayer, and the outer layer enclosing the liquid is called lamellar phase, which is similar to the plasma membrane. One end of the lipid bilayer, it is hydrophobic, whereas the other end is the hydrophilic. So functions looks like vesicles facilitates the storage and transport of materials in and outside the cell. It is even allows the exchange of molecules between two cells. Because vesicles are enclosed inside a lipid bilayer, vesicles are functions in metabolism and enzyme storage. They allow temporary storage of food and also the control the buoyancy of the cell. So here you have your plasma desmata on the left side. So this is the 3D version of it, some cross section. So you have here a cytoplasmic sleeve in between it, and these are central activities cavity and arm-like protein filaments. And here you have desmotobule. These are cell walls with adjacent cell walls. Here is the middle lamella. Then in the center is a central rod, and here is smooth endoplasmic reticulum. These are structures present in plant cells only. So they are the tiny passages or channels that allow the transfer of materials and communications between different cells. There are around 103 to 105 numbers of plasmodesmata connecting to two adjacent cells with the 50 to 60 nanometer in diameter. So plasmodesmata has three layers. It has continuous with plasma membrane and the same phospholipid bilayer, and the cytoplasmic sleeve that is continuous with the cytosol that allows exchange of materials between two cells, and desmotubule, which is a part of the endoplasmic reticulum that provides a network between two cells and allows the transport of some molecules. So plasmodesmata are the primary site for the communication of two cells. It allows the transfer of molecules like proteins, RNA, and viral genomes. So this is how your various plastids looks like. This is your proplastid. From this the etoplast is made. Then the chloroplast, chromoplast. For the leucoplast is made from protoplast. Then from the leucoplast your amyoplast, eleoplast, proteinoplast. Various structures are made in the case of plastids. So plastids are the double membrane bound structures present in plants only and involved in the synthesis and storage of food. They are usually oval or spherical with an outer and inner membrane between which lies in the intermembrane space. The inner membrane enclosed a matrix called stroma that contains a small structures called grana. So each granum consists of several sac-like thylakoid piles, one of the each connected to stemma lamella. So mainly functions they are involved in the storage of food and primary starch, and also involved in metabolic activities including photosynthesis as it contains enzymes and other components required for it. Then comes your, I think last not the least, vacuoles, which has tonoplast, central vacuole, cell sap. So vacuoles are membrane-bound structures varying in size of different organisms. The vacuole is surrounded by membrane called tonoplast, which encloses fluid containing inorganic materials like water and organic materials like nutrients and even enzymes. So these are formed by fusion of various vesicles. So vacuoles are very similar to vesicles in structure. So functions are act as a storage for nutrient as well as a waste material to protect the cell for toxicity. They have an essential function of homeostasis as it allows the balance of pH of the cell by influx and outflow of H plus ions to the cytoplasm. And vacuoles contain enzymes that play an important role in different metabolic processes. So that's it, student, for this type of principle of genetics in the case of ultrastructure of cells and cellular organelles. So I can share this part with you now. So I have two lectures, so I will share both with you. One is the larger one is the smaller one. So you can choose any of both for your studies. So I will share here. Today is the 6th day, right? So I have also another one from the syllabus that is a subjective one, but it has a lot of context. So in that reason I didn't took this part, because this will take me, if I started to read for you, it will take me zillion times, and it's not a presentation type of slides. So these slides are made for your examination point of view. I still have them, so I'm sending you these slides also. So go through them, so you will also have the same content but in a larger way, in a bigger way. So my reason is here to just to give you uh, a gist of everything what we have done, but please go through them once also. So on here I will take a 5 minutes break and then see you at 40 at 40 at 40 8:40. Yeah, so for the next lectures, so I will take tomorrow's lectures today's also, study of chromosomes. this will be your next lecture. So, class, welcome to the second session of today's project of our principle of genetics. So, I will be teaching here the chromosomes. We will go a little bit more deeper with the chromosomes, how chromosome works, how various histone, octamer, nucleosomes, nucleolus, how all these are established, how the architecture looks like inside the inside your chromosome. So this is the appendix. So we will be dealing with the prokaryotic chromosome, chromatin structure, eukaryotic chromosomal structure, and the flow of genetic information. So we will be all dealing with this part today. So prokaryotic chromosome structure, that is E. coli chromosome. It is a single closed circular DNA with 4.6 mega pair, and it is packed into a region called nucleoids, contain high concentration of DNA, and as well as the proteins called with the associated with the DNA. It has continuous replication, so no cell cycle is happening in this case. So this is how your DNA of your prokaryotes looks like. It's like spindle fiber shape. Some are not spindle. So these are supercoiled domains, and inside is the protein membrane or core or scaffold. Then is there's 50 to 100 kilobase per independent loops of domains. Observed under these were the observed under electron microscope. So there are 50 to 100 kilo domains or loops per E. coli chromosome with 50 to 100 kilobase per loop. So end of loops are constrained by the binding of structure, which probably consists of proteins attached to the cell membrane. The supercoiling is highly negative, and individuals' domains may be supercoiled independently, and direct biochemical evidence is lacking for different levels of supercoiling in different domains. So DNA binding proteins, prokaryotic chromosome structures, histones like protein essentials for DNA packaging to stabilize the constraint and supercoiling. And HU is a small basic dimeric protein, non-specific binding to DNA, most abundant. And HNS, it's a neutral monomeric, partially non-specific binding. And site-specific, there's a DNA binding proteins important for the organization of the DNA domains. So now comes the next part in eukaryotic cells, how your chromatin is adjusted. So this is the major structure in DNA compaction. So DNA nucleosome. So here you have a DNA which is compacted to the nucleosome with the help of histones added to that. So these histones they make beads of a string of fiber 10 nanometer. Then there's a 30 nanometer fiber from that they make, and then from that a metaphase chromosome is made like this. So that's how from your DNA compact to full metaphase chromosome with 30 nanometer fiber is made like this. So this is how your in interface your chromatins is in diffuse state, but as soon as the mitotic phase starts, so it goes from metaphase to telophase with the separation of two different cells in cytokinosis. So here your chromosome is condensed, here it is diffused. So this is the short region of DNA double helix you can see here on the top in 2 nanometer. Then they are arranged on the chromatins like this, dumbbell shape. They are called twice. And then 3 nanometer chromatin fiber of packed nucleosomes with 30 nanometer, and then section of chromosome in an extended form, that is 300 nanometer. Then further condensed section of metaphase of chromosomes exists. Then further entire metaphase chromosome come into this section like this one, 1400 nanometer. So major protein components of chromatin, four families of core histones H2A, H2B, H3, H4, and additional non-core histone proteins H1. The small at 10 kilodalton for core histones and 23 kilodalton for H1. Basics and tightly binds to DNA. So here you can see there is H3 and H4. They make together dimer H3 and H4, and H2A and H2B. They together makes H2A and H2B dimer. So together these two dimers they make a tetramer, that is H3 and H4. And with this tetramer with H3 and H4, and with this dimer with H2 and H2B, it's make a histone octamer. And this is a complex histone with DNA. So this is how your H3 H4 tetramer plus H2A H2B dimer, they come together and act as a whole octamer, histone octamer. So remember this part. So this is the histone octamer. You can see here at the one side DNA enters, and then other side it's leaves. So this is the top view, this is the side view of the same. Then this is the D2 chromatin structure, nucleosomes, which is the basic unit of chromosome structure consisting of a protein octamer containing two each of 4 histones, with 146 base pairs of DNA wrapped 1.8 times in left-handed fashion around it. Wrapped of DNA into nucleosomes accounts for the virtually all negative supercoiling in eukaryotic DNA. And further, this stabilizes the points at which DNA enters and leaves the nucleosome. So nucleosome is the reason your everything is stabilized. So this is a C tail of H1 stabilize the DNA between nucleosome cores. This is shown in a different diagram. Same thing. So you have your nucleosomes on the side, nucleosome here, and here is the histone H1, and then your DNA is coiled around it, and linker DNA is in between them, which is of 10 to 100 base pairs, average 55 base pairs. And these nucleosome repeats are around 200 base pairs, so with the co-linker DNA. So additional DNA required to make up 200 base pairs, nucleosome repeats around 55 base pairs. So then these axes of coil, they are high ordered, left-handed helix and nucleosome per turns, 6 nucleosomes per turns. So that then they started these nucleosomes, they started to make a chains of turns here, like 6 nucleosomes together. So then after they're making these chains, so your chromosomal DNA looks like in this loop shape. Chromosomes, your DNA inside the chromosome, your DNA looks like this in loop shape with 300 nanometer to 30 nanometer of size. So here's your mitotic chromosome. Here you can see telomere on the top. These are your sister chromatids. This is your centromere in the center, and these are your chromatids. Yeah. So when you check them on the other side, other way around, from a sectional point of view, cross-sectional point of view, here you can see loops of fiber with 30 nanometer fiber, nuclear matrix, and chromatids. So nuclear matrix loops of this is how your mitotic chromosomes looks like at the metaphase state. Then comes your mitotic chromosome. So these are done with the dye. You can see them being separated during the phase. And this is the centromere. Sorry. So this is so we are talking about eukaryotic chromosome structure so far. So this is the centromere in the center. Yeah. And this is third one is the short arms, the fourth one is the long arm, and this one hole is called as chromatid. Okay, so region where two chromatids are joined, this is known as centromere, sites of attachment to the mitotic, spindle and kinetochore, and this is your centromere DNA. Then comes your eukaryotic chromosomal structures, that is your telomeres. These are specialized DNA sequences which forms the ends of your linear DNA of eukaryotic chromosomes, contains up to 100 copies of short repeated sequence synthesized by the enzymes telomerase independent of normal DNA replication. A telomeric DNA forms a special secondary protein to protect the chromosomal ends from the degradation. So this is how your quadruplex form of human telomeric DNA looks like from the top point of view. So in the interface, the chromosomes have got much more diffuse structure. Their chromosomal loops remain attached to the nuclear matrix, cannot be visualized individually. So like this in the interface state. Your your telomeres are on the top, and this is how your chromosomes looks like in that case. Then comes your next part of your chromosomes, that is heterochromatin and euchromatin. Heterochromatin is a highly condensed materials, transcriptionally inactive, can be repeated a satellite DNA close to the centromeres and sometimes a whole chromosome. Euchromatin, chromatin other than heterochromatin, more diffuse and not visible, region where transcription takes place, not homogeneous, only a portion. Euchromatin is a transcriptionally active, where 30 nanometer fiber has been dissociated to beads on a string, and parts of these regions may be depleted of nucleosome. So this is your animal cells, this is your plant cells. So in both of them, here you can see there is a nuclear envelope, then there is an inner membrane, then there is a nucleolus, then there is a nucleoplasm. These are ribosomes and nuclear pore. But here is your heterochromatin present outside your attached to your nuclear membrane, whereas your euchromatin is inside of this. This is inside of that. So this is showing the human cell, showing the location of heterochromatin, where your heterochromatin is present. Then there is a DNA's one hypersensitivity. These are the active regions of chromatins where 30 nanometer fiber is interrupted by the binding of specific proteins to the DNA, ongoing transcription, and are characterized by the hypersensitivity to deoxyribonuclease. That is DNase one, like this. So this is CpG methylation. Methylation of C5 in the cytosine base of C5CG occurs in mammalian cells, signaling the appropriate level of chromosomal packing at the site of expressed genes. CpG methylation is associated with the transcriptionally inactive region of chromatin, and their islands surrounded the promoters of housekeeping genes responsible for epigenetics and may also to the RNA silencing. So these are your CpG islands present on the chromatin chromosome, which are acting like a housekeeping genes, and these are methylated CpGs, and these are unmethylated CpGs, promoter of the housekeeping genes. Uh, then there are also variants of your histones, that is H3 and H4. We have talked about H2A and H3 are also modified one. So histone modification can occur in the biological processes such as that gene regulation, DNA repair, and chromosomal condensation. So how the flow of genetic information happen, as we have known that it happened from DNA to RNA and RNA to protein, and this whole method is known as your central dogma. Sometimes DNA could self-replicate to produce a DNA with the help of DNA polymerase, and then with the help of transcription, RNA polymerase it's produce sense mRNA. With the help of transcriptase, it could produce reverse transcription leading to production of DNA, and further translation can happen with the help of ribosomes to produce proteins. And RNA could also replicate and produce RNA dependent RNA polymerase with the help of RNA dependent RNA polymerase. So making a positive sense and negative sense of RNA. So this is uh, a common summary of that thing, DNA to RNA to protein, the central dogma. So how the flow of genetic information happens? So it's mainly happening because of you can see there's a promoter, there's a transcribed region, there's a terminator. There is a transcription RNA polymerase. This is RBS. This is stop codon. This is start. This is here is start codon AUG, and this is how your then afterwards RBS is attached ribosome, and you have amino acid tRNA is present, and proteins are produced in the case of prokaryotic region. In the case of eukaryotic gene expression, with the help of RNA polymerase 1, first you have your exons and there's introns. Splicing is done, RNA processing is done to remove the introns, only the exons are left. And then they make a polytail of poly A tail. Then the translation happens in the presence of ribosomes and amino acid transferase, tRNA. Then a protein is produced in that case. So this we have discussed all various parts of today: mitochondria, chloroplast, Golgi apparatus, lysosome, cytoskeleton, ribosomes, vacuoles. So that's it for today. I will share this one also. The thing. This message was deleted. What was the message? So day 7 workshop. And also student, there's a topic of mitosis and meiosis, so I'm also sharing this presentation, but we have done that earlier in our practical classes. Understand with the basics mitosis and meiosis. um, so yeah, so we are done for today and tomorrow. So tomorrow there will be no class, as we are done today. uh, so my streaming will not work. Something goes wrong with my streaming. So here I have to say good night to everyone. Uh, I see you. Uh, okay, my streamings got saved. Any questions? Anything else, students? Okay, then on that note, thank you very much for today. It was pleasure meeting you all, and I see you tomorrow. So we are done with almost our unit one is finished. Uh, so so. So if you see from the unit point of view, so we are done with all these things already. Ultrastructure is also done. Study of chromosome structure is done. The karyotype and ideogram I will do it at the end of our lectures. So that we will do, and also we'll do the CIB techniques, that part also then with you. So these two parts are left. So from next onwards, so we will see us on today is Monday. I will see you on Wednesday now. So tomorrow will be no lecture. So from Wednesday onwards, we do this DNA and RNA structures, transcription, and yeah. So leftover things we will finish it off with it. So not much left now. So the unit two is left. So unit one is complete in day 7. So with 3 days, I hope we are able to finish this part also. We will. We will. So okay. Yes, the class will be day after tomorrow. That is Wednesday. So today is 21st. On 23rd will be the class. So I have to leave out of station for tomorrow, and I will be back day after tomorrow. So I try to compensate your two lectures today together, with ultrastructure and chromosome together. So they were big topics. So but we try to finish them today together. Okay, all clear then. I will say goodbye. Have a nice time, and thank you very much. Keep working on your review also, because don't get slow, because it takes some time sometimes. Uh, so on that note, thank you very much. It was pleasure meeting you all. Good night and stay safe.